this video, you'll have made 120 million red blood cells. Red blood cells, platelets, and white blood cells are the major types of cells that circulate in the blood. These cells play key roles in providing nourishment, in fighting disease, and maintaining homeostasis. In the Forsberg Lab, we study hematopoiesis, which is the process by which blood cells are formed from hematopoietic stem cells. By studying hematopoiesis, scientists and clinicians can better understand the processes behind blood disorders and cancers. The goal of the Forsberg Lab is to understand hematopoietic stem cell fate decisions throughout life. The entire immune system is generated from HSCs, or blood stem cells. I'm specifically interested in how maternal exposure to pathogens influences seeding of the immune system from HSCs during fetal development. For example, my current project focuses on how maternal immune activation affects the predisposition towards asthma in the developing fetus, which likely everyone watching this video probably knows someone who has this disease.
These HSCs that are established during fetal development become slightly different when they reach the adult stage. Two of these differences are their ability to self-renew and mature into diverse blood cells, two very powerful capabilities that are harnessed in stem cell transplants to successfully regenerate the blood of the recipient. However, there remains a high clinical demand for suitable donor HSCs. My goal is to understand the mechanisms that control the regenerative capacity of fetal HSCs and their persistence into adulthood. And understanding these mechanisms will improve clinical utility of hematopoietic stem cells.
Epigenetics describes the mechanisms of how genes are turned on and off. This includes small changes that occur on individual genes as well as large changes that occur on the entire nuclear landscape. In the context of blood development, I'm interested in how these both large and small changes affect the blood stem cell's ability to make decisions as to which type of mature cell it wants to become. Understanding these mechanisms can allow us to one day be able to make efficiently artificial blood.
In the aging blood, we made an exciting discovery that there is a unique change in the stem cell's ability to become platelets. And I predict that this age-specific pathway is what contributes to increased risk for platelet disorders in the elderly. Therefore, my goal is to understand the molecular mechanisms that control platelet dysregulation during aging. And by defining novel regulators of platelet formation and function, my studies will reveal new strategies for improving the health of the aging population.
We tackle these goals from multiple angles by in vivo and in vitro experimental approaches, by focusing on specific molecules as well as analyzing global changes. Ultimately, we want to understand the molecular determinants of hematopoietic stem cell fate decisions so we can prevent and treat both genetic and acquired disorders of the hematopoietic system. Your contribution to these projects will support graduate student-driven research in the Forsberg Lab at